Continuing on with the special senses, now we get into the physiology of taste, and we get into a little bit more complex, anatomy and physiology, form and function, as we discussed these uh, last four special senses. Taste is known, we talk about the term gustation, you'll see. Gustatory sense, again, involves chemoreceptors stimulated by various chemicals that we put in our mouths. Normally, it's something that we eat. Obviously, it doesn't have to always be something. It could be something you drink. It could be something, a medication that you take, you taste it. It is a complicated process involving your olfactory chemoreceptors, the nose. That's why we started off with that. Thermoreceptors, when you put something in your mouth, you know whether it's hot or cold. You also have nociceptors in addition to your gustatory chemoreceptors. So it's a lot going on. We smell food, we know the temperature of food that we eat or something that we put into our mouths, taste of it, all of that plays a role. All right, so it begins with a stimulation of specialized receptors called taste buds. Everybody's probably familiar with taste buds. Stick out your tongue, look in the mirror, and you see these small little balls on your tongue if you look at it really closely. All right, so taste buds are small clusters of receptor cells and supporting cells scattered about the tongue and other surfaces of oral cavity. Each taste bud is associated with sensory neuron that carries information to the central nervous system, CNS. All right, so let's study the anatomy. Taste bud is a tongue is covered with this papillae, these round projections, which can be classified on shape. You have different classifications of the size of these taste buds. Valate or circumvallate papillae, largest of the four classes, dome-shaped, contains hundreds of taste buds. Fungiform, mushroom-shaped, contains few taste buds. Foliate, ridges on the sides of your tongue, only contain taste buds in childhood. Filiform, long, thin cylinders scattered across tongue, don't have taste buds, but contain sensory nerve endings, detect food texture and temperature. Right? Taste buds, usually located on the lateral surface of the papillae, contain 3 cell types, right? Obviously, you have your gustatory cells, the ones that actually do the tasting, specialized epithelial cells with microvilli project into small opening papilla surface, taste pore, display receptors, detect different tastes, right? And this is associated with your sensory neurons that carry taste stimuli to CNS via your cranial nerve 7, cranial nerve 9, and cranial nerve 10. You also have basal cells, stem cells, that differentiate into new gustatory cells, since their lifespan is only 10 to 14 days. And then you have supporting cells. So similar structure to the olfactory. Your supporting cells surround them and support your gustatory cells. And so this is what we're looking at. As you can see here, we have the tongue. It shows you some of the different, it shows you where the different taste buds, different shapes of taste buds are located on the tongue. Foliate on the sides, fungiform towards the front, anterior, superior section, the velate. You see more in the towards the back. And then it shows you the breakdown of the taste bud. Here we have sensory neurons. Right? We're going to talk about the transduction of the chemical and the chemicals into a signal that can be sent for taste. And then it shows it even more closer detail here. The microvilli, basal cells, which differentiate into new cells that'll sense taste, supporting cells, gustatory cells. Okay. So let's talk about the physiology of taste. How does a food go into our mouth? For example, we eat it, we taste it. A signal is sent to our brain, and we know, okay, I'm eating a cookie, for instance. Even with your eyes closed, you can know right away what you're tasting, which is amazing. So the sense of taste involves signal transduction. So you're converting a chemical, a food, for example, into an electrical signal that can be sent to your central nervous system. Taste sensation relies on detection of 5 classes of chemicals. We have sweet taste, simple sugars, glucose, fructose. We also have sour taste produced by hydrogen ions, citric acid and lemon juice, for instance. If I drink lemonade, I know that's sour. Salty taste, elicited by presence of metal ions, sodium and potassium, particularly sodium. Salt is NaCl. We have bitter flavors produced by nitrogen-containing compounds. We have umami, taste associated with meat or broth, produced by glutamate or other amino acids. So our body can differentiate between all of these different chemicals, right? Hopefully that's starting to make sense. A little quick input here. Are you a super taster? Most taste preferences are determined by foods eaten while growing up. When asked to taste a specific compound, 20% of the population tastes nothing, 50% find it mildly bitter, and 25% intolerable. They're considered super tasters. Super tasters are people who have a heightened sensitivity, and it's thought to be something that's genetic. And they have an above average number of the Fujiform, on their tongue, towards the anterior portion of their tongue. Those taste buds. All right. Let's continue with the physiology of gustation. Effect of taste begins when the chemical, so the food that I'm eating, binds to the receptor on microvilli of the gustatory cells. Right. So what I'm eating, that chemical binds to the receptor. Taste receptors classified by the substance they detect. There's only one type of receptor associated with individual gustatory cell. Right. So we take different cells, taste different things. Activation of taste receptors. Substance must first dissolve in saliva. That's one of the roles of saliva. Sort of like when the odorant has to dissolve in the mucus in the nose, the food we eat has to dissolve in the chemical. The food we eat has to dissolve in saliva before it can reach the taste bud, where it may be detected as a gustatory stimulus. Then we have changes in ion movements. Depolarize the gustatory cell's plasma membrane, right? Depolarization happens based on this chemical. Opens voltage-gated calcium ion channels, and that produces an action potential, right? So here we have the diagram. This is example the transduction, changing of a salty taste or sour taste in the gustatory cell to an action potential. All right, so this is so changes in ion movements, depolarized gustatory cells, plasma membranes. So you have this specialized gustatory cell, which the chemical reaches the microvilli. The gustatory cell, specialized cell, depolarizes. Depolarization of this membrane opens voltage-gated calcium. Calcium enters the cell. Voltage-gated calcium channels. Calcium enters the cell. The calcium triggers the release of the neurotransmitter, which produce an action potential and the axon of the sensory neuron. So the specialized gustatory cell tasting, in the taste bud, taste cells, this cell can depolarize. Notice it's not a neuron, but this cell depolarizes, sends a signal to the neuron, and then that neuron depolarizes and sends a signal. And now we've converted the chemical of taste to a signal that can be transmitted to a neuron, and that signal eventually sent to the brain. So the pathway as taste stimuli travels to various regions of the brain for processing, just like in the olfactory. So axons of facial, glossopharyngeal, vagus nerves carry taste stimuli from the tongue to the CNS, terminating in solitary nucleus in medulla by synapsing on central sensory neurons. Axons, so it all comes together. Axons from solitary nucleus synapse on neurons in the thalamus. So this does go to the thalamus, unlike the signals coming from the nose. This goes to the thalamus, and the thalamus sends impulses to the primary gustatory cortex, which is in the cortex of your cerebrum, in the parietal lobe. So your taste signals travel to either the inferior region or frontal lobe for integration. After the parietal lobe, integration with visual and olfactory information, or to the limbic system, provides emotional reactions to taste. All right, so we can take something that we taste and combine it with something that we've seen. I see the lasagna as I'm tasting it, so that once I taste it, that information can communicate with information that I've seen. Or once I taste something I've heard cooking, that communication, that information can work in conjunction with what I've heard cooking. So the frying of fish, for instance, I can associate that with a certain taste. Remember, all of this is working together in the same brain. That's the amazing part about it. So much so, when I get to the point to if you've ever gotten to the point to where something can be cooking and you can taste it before you taste it. So like fried chicken is cooking and you can know the taste in your mouth, even though you're not actually eating it at this time. Just from smelling it or just from seeing it, you can almost taste it. Limbic system, brain working together, different parts, your brain combining what you see, what you hear, what you smell with what you taste. Right, so that's just showing you there the vagus nerve, glossopharyngeal nerve, facial nerve, to the thalamus, to the primary gustatory cortex. Now let's go over the eye. All right, so the eye is a very complex organ. The eye eyeballs are located in orbits on either side of the nasal cavity, or on this right beside your nose. Each eyeball occupies the interior 1/3 of the orbit. Adipose tissue provides protective cushioning. That's why if you get like pushed, you can push on your eye a little bit. Don't do that, but you do have some adipose, some fat that helps protect it. You have a lot of accessory structures to the eye. You have eyelids, thin folds over the eye, cover the region of the orbit, prevent access by foreign objects, also distribute tears. Eyelids have the following anatomical features: tarsal plates, tarsal glands, modified sebaceous glands, secrete oil to prevent eyelids from sticking together. You also have commissures, where upper and lower eyelids meet. It's just showing you the structure of the eyelid. You don't need to memorize this, but just kind of interesting. You also have eyebrows and eyelashes. Hairs on the ridge of brow form eyebrows, prevent perspiration from running to your eyes, also can block glares. Eyelashes, everybody knows what eyelashes are. Have sensitive nerve endings, cause blinking when objects touch them, so they help protect your eye, reducing things from getting into your eyes. You have this conjunctiva, thin, continuous epithelial membrane, lines both posterior surface of eyelids and anterior surface of eyeball. Translucent, that means light can go perfectly through it. Translucent membrane in which tiny blood vessels can be seen. Palpebral conjunctiva covers eyelid inner surface. It turns back on itself, covers white part of the eyeball. You also have the lacrimal apparatus, produces and drains tears from the eye. The lacrimal gland in the superolateral region of the orbit posterior to conjunctiva releases tears and mucus into tiny ducts. Right? Lubricates, washes away debris. Lincoln sweeps tears immediately and inferiorly across eye surface. Drain into passages that lead to the nasal cavity. So we decide we kind of clean out our eyes. And this is just telling you the process of that. Okay, lacrimal gland there. Showing you how and then it drains into the nose, to your nasal cavity. Clean out your eye. And then we have extrinsic eye muscles. We do see these muscles that help the eye move. All right, 6 muscles originate from the orbit wall, insert into outer layer of the eyeball. Superior, inferior, lateral, medial rectus extend from common tendinous ring on posterior wall to their respective insertion sites on the eyeballs. We have these, 6 muscles that basically can help us move the eye. Each rectus muscle moves eyeball primarily in direction for which muscle is named. All right, so we can, the eyes can move all directions, which is great, so we can see things all around us. Okay. And then we also have persistent movement of the eye require a great number of small motor units, consisting of 2 to 3 muscle fibers. 3 cranial nerves provide, this is where we get to some important stuff. 3 cranial nerves provide an innervation to 6 muscles. So now we have your cranial nerve 4, 6, and 3. Innervates superior oblique, innervates lateral rectus, innervates remaining 4 muscles. So you have 6 muscles in the eye 3 cranial nerves help with the movement of those 6 muscles in the eye. The term lazy eye, strabismus, disorder present at birth, eyeballs are not properly aligned with one another, can sometimes lead to double vision, all right? Okay, this is the way you can remember which muscles are innervated by which cranial nerves, right? It's a mnemonic device, uses chemical equation and analogy, LR6, SO4, all the rest are 3. So cranial nerve 4 innervates superior oblique, SO4. Cranial nerve 6 innervates lateral rectus, LR6. Cranial nerve 3 innervates remaining 4 muscles. Right, so we've covered 1 cranial nerve with the nose, 3 with the tongue, and now another 3 with movement of the eye. All right, let's get into the eyeball. This is where things get a little complicated. The eyeball, hollow sphere, consists of outer wall that surrounds several chambers, supports lens, focuses light as it enters the eye. We have 3 distinct layers: fibrous layer, vascular, and neural layer. Talk about. And at the eye, we do a wonderful dissection in the class normally, which we take apart the eye. It's a very amazing structure. Nose is quite simple. The tongue is actually pretty simple, even though taste is, all of these senses are important. The eye it's a pretty complex structure, as you see. Fibrous layer, vascular layer, neural layer, sort of here. And so the idea varies, remember, always think big picture. The idea is we want to basically take light, that's the stimuli, light, and convert it to a signal that can be sent to our brain. So transduce it to a signal that can be sent to our brain and eventually interpreted by our brain. And we can say, I can look at this screen and see PowerPoint presentation. I can see the computer, I can see the letters, and my brain is figuring all that out as the light is reflected off of that into my eyes. Special senses are incredible. All right, so let's go through this. The fibrous layer, outermost layer, we have what they call the sclera, white part of the eye, covers nearly entire eye. It's made of irregularly arranged collagen fibers, helps resist deformation from external and internal forces, so it maintains the shape. Your eye needs to keep a certain shape so that when light enters it, the light comes in the right way. We're going to talk about it so it can get transduced into an electrical signal that can be sent through the optic nerve. Cornea, continuous with the sclera anteriorly, translucent, light goes right through it. Translucent due to parallel arrangement of collagen fibers. It is avascular, doesn't have its own blood supply, allows light to pass into the eyeball. I see the light. All right, so sclera on the outside surface. Then we have the cornea, which is continuous with the sclera, allows light to pass into the eyeball. Vascular layer, middle layer, lies beneath the cornea and sclera. Then we have the choroid, most extensive component. That's where we have capillaries, blood supply, also pigment. Oh, she got blue eyes. I think she has brown eyes. Minimizes scattering of incoming light rays. Remember, we want the light to come in and focus. We also have the Ciliary body, continuous with the choroid body, anteriorly, contains smooth muscle that surrounds the lens. Some suspensory ligaments, allows for contraction and relaxation, changes shape of the lens to focus the light so we can know what we're seeing. We have the iris. Everybody's probably heard of that. Colored region of anterior eye. This is what we think of when we think of eye color. Extension of vascular layer, amount of pigment, melanin determines iris color. Remember, melanin also determines your skin color. And then we have the pupil opening. That's the center of the iris, and that's where light enters the eyeball. All right, so this is that hole. So pupil, that's the opening. See the lens here. Suspensory ligaments hold the lens in place. Ciliary body. Choroid. All right. Basically, all of this is helping to light to come in and to focus the light. We also have papillary sphincter muscle in the iris contracts during parasympathetic stimulation. Remember parasympathetic and sympathetic? Reduces size of pupil. Can also restrict the amount of light entering the eyeball. And you have the reverse papillary dilator muscle. Contracts during sympathetic activation. Allows people to increase in size, more light. So we want more light in when we're in, remember, parasympathetic, rest and digest, right? We need less, when we're resting, we need less light coming in the eye. Sympathetic is when we're fight or flight. We need more light coming in the eye. We need to be more aware of our surroundings. Right, so increase light intensity, decrease light intensity. Parasympathetic control, sympathetic control. Then we go into our neural layer, innermost layer of the eyeball, known as the retina. Incomplete layer, only deep to choroid, composed of two layers. We have the superficial layer, thin pigmented epithelium, reduces light scattering, nourishes photoreceptors. And then we have a deep layer, which is your photoreceptor cells and cells that form the optic nerve. That's going to go to the brain. Your photoreceptor cell detect and transduce light into an electrical signal. We have rods, black and white vision, and low light levels, and cones, high acuity color vision. So that's how we see our black and white, and how we see our color. We also have several other cell types in this layer, including those that give rise to the optic nerve. We also have the macula lutea, yellowish central region of the fovea, contains large number of photoreceptors. Fovea centralis, central region of the macula lutea, contains large number of cones, tightly packed together. This allows us to have detailed vision and to be able to focus on an object. Fovea, focus, think of focus. We have this term called macular degeneration, progressive loss of visual acuity, particularly in the center of the visual field. It may also cause visual distortion, change in color perception. And we'll talk about that more in a little bit, but obviously everybody knows your eyesight degenerates, especially generally as you get older, but it can degenerate for other reasons as well. In the neural layer, we also have the optic disc. It's where the axons of the optic nerve exit from the retina. Does not contain photoreceptors, does not capture visual images. This is the blind spot. We also have the layers of the retina are in contact with each other, but are not physically linked by desmosomes or tight junctions. All right, so this is the fovea centralis, macula lutea, and the optic disc or blind spot. This is the way you can test you can look at this slide and test your blind spot. So you can read these instructions and test it. The lens, slightly flattened sphere. It's going to be very important behind the pupil and the iris. Pupil, the gap, and the iris. Focuses light on your retina from objects near eye. The ciliary body connects to lens by suspensory ligaments. Lens contains cells, lens fibers that lack nuclei, tightly packed and are translucent. Light can pass through them. So cells that lack nuclei, that means they're not going through any type of mitosis, tightly packed and translucent. And that's your structure of your eye. What are cataracts? Basically a clouded lens, and one of the most frequent causes of blindness. And they can come from different ways: trauma, exposure to UV radiation, diseases. Cataracts cannot be reversed, so you can't. It doesn't just go away. What they can do, though, is replace the entire lens, or they can give you glasses or contacts. Finishing with the eye, we have cavities and chambers of the eye. The eyeball is divided into anterior and posterior cavities by the lens and the ciliary bodies. Posterior cavity, larger cavity behind the lens, filled with a gelatinous material. Your eye is not hollow, so it's filled with this like jello. That's why we like to do the dissection, so you can see this for yourself, which is called the vitreous humor, made mostly of collagen and water. Helps press the retina against the choroid, maintain the eyeball shape. Then you have the anterior cavity, which is in front of the lens, towards the front, and ciliary body, which is divided into anterior and posterior chambers, both filled with the aqueous humor. Aqueous humor is a watery fluid secreted by the ciliary body, flows from posterior chamber through pupil into anterior chamber, drained from anterior cavity into sclerovenous sinus, which is a blood vessel network at the anterior edge of the iris. Right, so that's just the two different chambers. You have fluid in the front, a jelly-like substance in the back, and that is the structure. All right. Lens, cavities, chambers of the eye. We also have glaucoma. So your aqueous humor, when it cannot drain, the fluid in the anterior portion of the eye builds up, and the anterior and posterior chambers in the front of your eye, raises pressure inside the eyeball. Elevated pressure pushes on the vitreous body, compresses and damages your retina. It can cause blindness, which is not good. A symptom of glaucoma can be a gradual loss of vision, right? So another issue with the eye. So how do we see? Vision is the perception of light reflected by various objects. So light is reflected into our eyes. Eyes and visual pathways in CNS can determine an object's shape, size, and color, distance, radius, how fast it's moving, direction of movement. What are the principles of light? You don't need to know these in so much detail. This is more physics. Some energy exists as electromagnetic radiation, different wavelengths, gamma rays, X-rays have short wavelengths, microwaves, radio waves have longer wavelengths. Visible light has a wavelength that the human eye can detect. Shorter wavelengths of visible light are blue and violet, longer wavelengths are red. The basic unit of light is called a photon, and it stimulates photoreceptors in your retina. Those photoreceptors will then help start the process of transducing the light that you see into an electrical signal that can be transported to your brain, just like all the other special senses. Very similar. So this is just showing you the electromagnetic spectrum. We can't see gamma rays or X-rays. We can't see radio waves or microwaves, but we can see waves with the wavelength from 350 nanometers to 750 nanometers. That's called visible light. Light can be bent, which is called refracted when it passes through certain objects. Refractive index of an object measures the amount of refraction exerted on light. Air can refract light and by an index of 1, so it's not significant. Water definitely refracts light more. If you're seeing something through like a glass of water, a lot of times it can alter it significantly because water can refract light. Refraction of light also depends on the angle that light strikes the surface. Greater angle causes greater refraction. A convex lens surface that bulges outward causes light rays to bend inward. So a surface that bulges outward causes light to bend inward. Concave, thicker on the edge, depressed in the middle, causes light rays to diverge outward. When rays converge on one point, it's called a focal point. It's said to be focused. Which one do you think happens in the eye? Convex or concave? We want convex. We want it to focus. So our lens is going to be closer to this shape. To convex than concave. All right, so focusing light on the retina. Clear vision requires that light rays are focused on the retina, not in front or behind it. So 2/3 of the eye's refractive power occurs as light passes through the cornea, has refractive index close to water. Lens provides for fine tuning and refractive adjustment. So between passing through the cornea and then eventually through the lens, we get the refraction needed, right? Emmetropic state, eyes relaxed and focusing on distant objects. Lenses in normal, flattened shaped. Parallel light rays are minimally reflected by cornea and focused on the retina. The light rays from the objects, notice, so this is when you can see something in the distance, but not necessarily the things up close. Light rays from objects closer to the eye are more scattered. They need more refraction. Lens becomes more thickened, accommodation, refracts light more than flattened lens, so more light rays are focused on the retina. Right, so it's just showing you the concept. It's easier in general, when the eyes relax, to focus on something off in the distance, to see like I can see the door that I'm looking at, which is across the room, but my eye has to, if I look at an apple, as you see here, look at an apple in your hand, the rays are diverging when they reach your eye. A rounded lens provides the additional refraction needed to focus. Right? So this is just showing you how we can focus on certain things. Right? So off in the distance, my cornea can do enough of the refraction by itself to be able to focus it. But once it gets closer, my lens has to work harder to help refract what we see. Events that allow for your lens to shape change, your ciliary body, which we talked about. Surrounding lens relaxes when viewing distant objects. So your eyes are a little bit more relaxed when you view something in the distance. Pulls your sphincter-like smooth muscle away from the lens, creating tension on the suspensory ligaments. Flattens lens, reducing its refractive capability. So your body contracts when viewing objects nearby. So when I'm trying to view something close, my cornea my cornea alone can't do the necessary refracting. So my lens has to do some refracting. Allows for accommodation. Your ciliary body moves closer to lens and suspensory ligaments, slacken, allows lens to change to more thickened state, increasing refractive power. What we call pupillary constriction, limits the amount of scattered light that makes objects appear blurry. Enters edge of lens, objects appear more focused. Then we have convergence, process by which eyeballs move more medially to direct light rays onto photoreceptor dense region of the fovea. I'm not going to go through this concept boost right now. We do have some errors in our refraction. Limited accommodation due to aging lens or shape of eyeball. So as we get older, all of these things, sense of smell, sense of taste, sense of sight, all of these things are affected. Right? As the lens ages, it can't accommodate, it can't change shape as well to be able to refract the light as well, to be able to focus the light onto your retina. Therefore, it's harder to see as you get older. So near point of accommodation, closest point at which the eye can focus on an object, increases with age as lens becomes less flexible. So it's not as flexible. It's harder to focus. That's why you'll have people, when they're reading something, they won't put it here. They'll put it a little bit further away from them, or they will wear glasses, reading, what we call reading glasses. Okay. So your ability to see, your accommodation, the ability for your cornea, but more importantly, your lens to be able to focus light onto the back of your retina depends on its ability to have the correct shape. Okay. Farsightedness, you also have hyperopia. Eyeballs too short or cornea is too flat. That would also change the way we see things. Lens is unable to accommodate to focus light on the retina. Instead, focuses behind the retina, causing blurry vision when looking at close objects. So what we do is you get that person convex lenses that allow light to converge on their retina, right? It helps them if their lens can't cause light to converge enough to focus on the retina, we give them glasses that can help do that, as you can see here. Then we also have myopia, nearsightedness. Distance between the cornea and lens is too great, or cornea is too curved, so it focuses in front of the retina. For this person, we'll give them concave lenses, which makes sense. So it causes diverging incoming light, corrects that diverging incoming light before it contacts the lens. So it allows you to be able to focus it onto the retina, right? So light, it causes it to diverge a little bit more, and then your cornea and your lens can cause it to diverge and focus onto your retina, right? Nearsightedness. We also have astigmatism. Curvature of the lens of cornea is irregular. Light rays not evenly refracted. They have blurred vision at all distances. We have this LASIK, which now can be used to, this LASIK procedure, laser is used to remodel the cornea and smooth out any irregularities, right? So we can change the shape of your cornea and help you see better. Okay. All right, so how does this work? Cell types layered in the inner layer of the retina. Two types of photoreceptors, rods and cones, adjacent to outer pigmented epithelial layer of retina. Cones function best in bright light. Remember, we said the cones process color. The rods, more sensitive in low light, and as a component of peripheral vision. Your black and white. Your photoreceptors synapse with bipolar cells, which communicate with the retinal ganglion cells. Here we have cranial nerve number 2, retinal ganglion cells in anteriormost region of the retina, axons from the optic nerve, cranial neuron 2. Right. So now we've covered a bunch of the cranial nerves, your horizontal cells, and amacrine cells involved in image processing. And so this is your light hits your retina, retinal ganglion cells, amacrine cells, bipolar cells, horizontal cells, cones, rods, pigmented layer of your retina, and your choroid. A lot going on there. All right, here's the structural feature of the rod. Rod's cylindrical outer segments contain thousands of flattened discs containing pigment rhodopsin that absorbs light. All rods contain this pigment, do not distinguish between different wavelengths of light. Rhodopsin, composed of protein opsin and pigment retinol, derived from vitamin A. In the dark, your retinol is in bent configuration. All right, so this is showing you how we get from light. The idea is, what we're trying to figure out is how do we get from a refracted light to knowing that I'm looking at this door, for instance. All right, so the rod and the cones are a little bit different based on the light that they see. Your cones are outer segments contain iodopsin, composed of retinol and protein photopsin. Similar to opsin, but has slightly altered structure, allows it to absorb different wavelengths of light. Three forms of it allow response to wavelengths perceived as blue, green, and red colors. This is some neat diagram of that. Showing you a picture, micrograph. Transduction of light into electrical signal begins when the photon encounters disk in the outer segment of the rod or cone. In the absence of stimulation in the dark, so when you're in the dark, you can't see anything, eyes are closed or lights are off, photoreceptor cells are depolarized. Continuously release neurotransmitter into synapses with other neurons, reverse of most neuron activation. But in the presence of light, when we see something, when we're looking, when we're seeing something, photoreceptor becomes hyperpolarized and stops releasing neurotransmitter. Altering activity of neighboring retina cells sends information to brain. Okay. So in the dark, let's explain this. Let's go through this. Opsin, cis-retinol combine to form rhodopsin, disc membrane rod, G protein complex, transducin, blah, 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 photodiesterase enzyme are inactive, right? So all this is inactive. Sodium ion channels in the plasma membrane of outer segment are opened by second messenger, cyclic guanosine monophosphate, which is bound to them. Sodium ions flow down concentration gradient in the cell and depolarize cell. Okay. This is in dark. It's depolarized. But when light comes, when light strikes these photoreceptors, light converts the retinol into transretinol, separates from opsin, activates transducin, activates the PDE. Sodium ion channels close, photoreceptor hypopolarizes. PDE converts cGMP to GMP. Less cGMP reduces the number of open sodium channels. Influx of sodium ions is reduced. Right? Less sodium ions get in this case when light comes. Rhodopsin is bleached, so to speak. We'll talk about that. When transretinol disassociates from opsin, can no longer respond to light. Transretinol must travel to pigmented epithelial cells of retina's outer layer, where cis-retinol is reformed at the expense of ATP, requires energy. Reformed cis-retinol is then transported back to photoreceptor. So dark and light adaptation allow for adjustments in the amount of light present in the environment. So when light is suddenly reduced, cones can no longer function. Rods are slow to regenerate enough rhodopsin to function. Take up 40 minutes for rods to be completely functional. Retina is extremely sensitive. Like that's why if it was dark and I turn the lights on, all of a sudden, it kind of hurts. But you're very sensitive when you adjust to the dark. You can see you're trying to see anything. And the light, when light is suddenly increased, it bleaches the rods and the cones, resulting in a blinding glare. It almost hurts your eyes. Rods become non-functional. Cones can regenerate functional pigments faster, able to respond within a few minutes as the sensitivity increases. Color, you start to see color fast. Image processing by the retina in the dark. So photoreceptor depolarizes, releases glutamate into bipolar cells. It inhibits the bipolar cells, reduces its release of neurotransmitters. The retinal ganglion cell does not produce an action potential. No signals are sent to the brain via optic nerve. So we process the dark. This is how we are This is how we know we're in the dark. In the light hyperpolarizes photoreceptors, stops releasing the glutamate. Bipolar cells free from inhibition, depolarizes, releases neurotransmitter onto retinal ganglion cells. Retinal ganglion cell produces action potentials sent to the brain via the optic nerve. It's the process by which we see. Okay, so it just shows it here in the diagram. In the dark, retinal ganglion, bipolar cell, inhibited. Glutamate inhibits bipolar cell, reduces release of neurotransmitter. The photoreceptor depolarizes. Oh, sorry. And then the retinal ganglion cell does not produce an action potential. So we don't get this action potential. We know we're in the dark. In the light hyperpolarizes the photoreceptor. Bipolar cell is freed from an inhibition, depolarizes, releasing neurotransmitter, sends a signal. Retinal ganglion cell produces action potential, sent to the brain. We are seeing things. Notice the firing rate. Color blindness. Usually, we talked about color being with the cones versus the rods. So usually, if you have color blindness, you lack one of the genes for your one of your cone pigments. Missing a gene for red or green is most common. So people who are colorblind have difficulty distinguishing between the two. And here's some more details about that. 8 to 10% of males compared to only 1% of females. Color blindness has no cure. Most people learn to just compensate. And this is the way we can test for color blindness. If you don't see the 74, you only see the 21, then it may be an issue. All right, so what is the pathway? Detection and transduction of light. And we spent a lot of time on the eye, so we'll kind of go through this quickly. All right, so we have this visual field of each eye. So the amount that each eye can see is divided into right and left with a focal point in the center. Notice if I look at something, I don't see two doors, even though I have two eyes. So I have a focal point in the center. So that's showing you that. But I can see something with my right eye and my left eye if I close one or the other. So what's the visual pathway? So retina of each eye detects visual stimuli from portions of right and left visual fields. So my right eye can see some things on the left. My left eye can't see some things on the right. Some axons carrying visual stimuli cross the optic chiasma. So all stimuli from the right visual field are processed by the left hemisphere and vice versa. Your optic chiasma is a cross-shaped bundle of axons. Right here, see it here in the middle. Optic chiasma in the middle here. The cross-shaped bundle of axons of left and right optic nerves within the skull, just inferior to the hypothalamus. All right, so this is showing you your visual field. Left eye sees this side, the right eye sees this side, but the left eye can see some of the right side, the right eye can see some of the left side, and there's a focal point in the middle here. All right, so we have the optic tracts formed by the axons exiting the optic chiasma. So stimuli from the left visual field travel on the right optic tract and vice versa. So stimuli from the right optic field travels on the left. Then we have the lateral geniculate nucleus region in the thalamus. Remember the thalamus and the diencephalon. That's sort of like our control center, sends information, decides where the information is going to go. thalamus where majority of axons and optic nerves terminate, optic tract, terminate. Then we have optic radiation spread out deep to temporal and parietal lobes. Once they reach the primary visual cortex in your cerebrum, and the occipital lobe of your cerebrum, each half of visual field is represented entirely in opposite half of cortex. So what you see from your right eye and your left cortex, what you see from your left eye and your right cortex. Now the individual is consciously aware of visual stimuli. So your brain can decipher the shape, the color, the movement, location. And then you have integration with the different association areas. And you can fine tune that with eye movement. See here what we see, optic chiasma, thalamus, your cortex, and the occipital lobe. All right, consensual pupillary response, constriction of both pupils, response to light entering left or right pupil only. So some axons terminate in the midbrain, communicate with oculomotor nerve that innervate pupillary muscles in both eyes. When light is detected in one eye, pupil is constricted in both eyes reflexively to prevent damage, right? So if I see bright light, like even if I see it just with my right eye, both eyes will constrict to protect my eyes. We also have stereoscopic vision, perception of distance an object is from the eyes. This is very important, obviously, if you're driving a car. So your eyes face interior. Your eyes generally point towards the front. Much of the visual field for one eye overlaps with that of the other, right? Known as binocular vision. So remember I said my right eye doesn't just see the right side, it sees a little bit of the left side and vice versa. So they overlap a bit. So brain compares overlapping images to determine your distance an object is from the eyes. All right, so using both eyes, your brain can compare that information to be able to tell me, okay, how far is something away from me, away from my eyes, away from me. Image processing by the brain involves more than detection and awareness. Retinal axons project to lateral geniculate nucleus and primary visual cortex in your cerebrum, where the image from retina is mapped. You have two different pathways. Dorsal pathway leads to the parietal lobe. Ventral pathway ends in the inferior region of the temporal lobe, right? So more processing of what we see. With the dorsal pathway, we can interpret the motion if something is moving. With the ventral pathway, we can process the colors and forms of different objects. We also have many other regions of the brain that are associated with visual memory. Like when you're reading something and you're studying and you're trying to memorize it, it's going into your memory. So it goes to different parts of your brain to be able to help you to remember something or to recall, or you see something and it triggers a memory, which is also amazing. And this is just a diagram showing you the big picture of vision. So go over this on your own.